Yeah,
and I will now invite Tash Brock and shortly introduce her. So she's currently a PhD student at Rothamsted and working, as the title will show soon, on using CRISPR-Cas technology in order to improve wheat by doing gene editing or targeted mutagenesis there. So she's almost in her last year, and we met last year also for an AAB-PlantEd organized summer school, which we held in Düsseldorf. So Tash, I'm happy to see you. And thanks for agreeing as well for giving here a presentation. And I'm looking forward to your talk, and the stage is yours.
Great, thank you, Götz. So hi everybody, my name's Tash. I'm a PhD student at Rothamsted, and I'm going to be talking to you today about some of the work in my group on amino acid metabolism and my PhD project. So I'm going to be talking about two amino acids today, asparagine and lysine, and firstly I'm going to be focusing on asparagine. So why asparagine? Well, asparagine acts as a precursor to acrylamide formation. Now, acrylamide is a group 2 carcinogen, which is a food contaminant, and it's formed through the Maillard reaction. Now, over here is the entire Maillard reaction, and we're just focused on this bit down here where asparagine is being turned into acrylamide. Now, in terms of acrylamide in our diet, these graphs show cereal products in the EU. So we have the mean amount of acrylamide here, and the product with the highest amount is biscuits, crackers, and crisp bread. However, if we look at the graph over here, and we look at the contributions of acrylamide percentage in our diet, then bread is by far the highest. So this just illustrates that acrylamide is a very big problem and is massively consumed in lots of different cereal products. Now, it is a problem that's been known to industry for a long time, for many years, and we have made lots of advances, particularly in reducing acrylamide through processing methods, mainly by reducing cooking times and cooking temperatures without compromising on taste or flavor for consumers. However, all the kind of improvements that we've made in that area have been achieved now, and yet we still have very high amounts of acrylamide in different food products. Therefore, we need an alternative method for being able to reduce acrylamide further. And we have used CRISPR-Cas9 to do this. So this work was done by Sarah Raffan, who was a PhD student and postdoc previously at Rothamsted. And in her PhD, she designed 4 guide RNAs that targeted ASN2, which is asparagine synthase 2 gene. Now, she designed 4 guide RNAs that she then introduced into wheat via particle bombardment, along with the Cas9 gene and a selected marker gene, BAR. After successfully doing this and growing up the plants, she then analyzed them and identified 3 lines to take forward. This being line 178, which is an A genome null, and 2 total nulls, line 23 and 59. Now, interesting, line 59 also had edits in asparagine synthase 1 genes, as well as asparagine synthase 2. Having a closer look at these edits, most of them were deletions, as you can see here. However, there were some substitutions and single base insertions across each of the A, B, and D genomes, resulting in mostly dysfunctional truncated proteins. And when these lines were analyzed for their asparagine content, these were massively reduced and then also baked into different products like bread and biscuits to analyze their acrylamide content. So once this has been achieved in the lab, we then wanted to see if this could be repeated in the field. So we applied for permission to run the first CRISPR wheat field trial at Rothamsted, which has run between the years 21 and 25. And I'm just going to show you some of the results from this. So firstly, looking at the free asparagine content of the flour produced in the years 2022 to 2023, shown in the dark blue, and 2023-24, shown in the lighter blue. Now we have cadenza as our control. All of the edits in these lines was done in a background of cadenza. We then have our line 178, which is that A genome null, and then we have our lines 23 and 59, which is the total nulls. We then also have Claire and some tilling lines. Now what this shows compared with the cadenza, line 23 is up to 50% lower in asparagine, while our lines 59 were up to 5% of the condenser, which shows a massive reduction that has been repeated in the field there. But we're mainly interested in the acrylamide content. So we took that flour, made it into bread, and then toasted it. Here we have T0 is just untoasted bread, and then we have from T1 to T4 is each further minute of toasting, and analyze the acrylamide content for our cadenza in black, the line 23 in this dark purple, and line 59 in the kind of mid purple with Claire and our tilling lines. Now, as you can see over time, for each of them, the acrylamide content increases, which, as we know, is a Maillard reaction progresses. We produce more acrylamide. So this trend is shown here. However, when we look at the acrylamide content in both line 59 and line 23, compared with our cadenza, it is massively reduced. So in our T3 toast, line 59, the concentration of acrylamide was only 8% of that compared to cadenza, while line 23 was at 23% of the cadenza. And so the difference between these lines was highly significant. Now, this work has been done as part of AccuRed, another cost action, which Geraint mentioned earlier. So if you're interested in looking more at this, please go and check that out. Now, we also wanted to ensure that by producing this low asparagine, low acrylamide wheat, there was no yield effects or any phenotypic effects. So we have our yield on the left graph here and the 1,000 grain weight on the right, with both the years 23 in the dark blue again and 24 in the light blue. So again, we have the same lines here. We have cadenza on the left, and we have lines 178, 23, and 59 with Claire and our tilling lines. Now, in terms of yield, there's very little difference between our lines compared to cadenza here. But when we look at the 1,000 grain weight, there is a drop compared to cadenza with our edited lines. So the edited lines were producing more grains, however, they were smaller grains, but still within a normal range for wheat. And when we look at our tilling lines, there is a yield drop, a yield penalty compared with the Claire, but this could be reversed with more backcrossing. Now, during this time, during the field trial, the Precision Breeding Act was passed in England. And so once the editing was done and the transgenes using CRISPR are no longer needed, they can be segregated away. Because to be able to produce what's called PBO, precision bred organism, then these plants need to be free of foreign DNA. Now, in terms of our lines, we've confirmed that the line 23 is PCR negative, all of the transgenes, and confirmed by whole genome sequencing. And we're also screening our line 59 plants as well. Now, we are part of the Probity project, which is aimed at helping to commercialize these products. And they are currently being, the seed is being bulked up at farms across England to produce up to 100 tons, which can then be used in real world food pilot plants. There is also another project at Rothamsted led by Pete Eastman, which is part of the Probity project. This is all Innovate UK funded. And he has a barley, which is CRISPR edited to be higher in lipid content. And he has also been given the UK's first precision bred organism marketing notice, which is really exciting. So please go and check that out if you're interested. Now the EU is also slowly changing their stance on genome edited crops, or new genomic techniques as they call it. So we are going to have to see what they say on their new regulations and how that lines up with the Precision Breeding Act in the UK. And we'll have to see if their definitions of PBO line up. So I'll have to keep an eye on that for the future. So now I'm going to switch tacks a little bit, and I'm going to talk about my work, my PhD project, focusing on lysine. So why am I interested in lysine? Well, lysine is one of our essential amino acids, which means humans cannot make it, and we need to get it in our diet. Now, animal products and meat are a great source of lysine. However, people who choose to live a plant-based diet, or people in less economically developed countries who do not have access to sufficient quantities of meat, are at much higher risk of lysine deficiencies, because wheat, like many cereals, is low in lysine. Now, lysine deficiencies can cause weakness, fatigue, a stunted mental and physical development, particularly in children under 5, and can also lead to having a weakened immune system, which is going to make you susceptible to lots of other health conditions. Now, lysine deficiency doesn't just affect humans, it also affects any monogastric animal, including pigs and chickens. And so when farmers are feeding their animals sort of low quality, low protein wheat, they're going to have stunted growth and development. So to combat this, farmers have been importing soybean, which is high in lysine. However, this is really damaging to the environment and also damaging for the UK animal feed industry. So we want to try and tackle these problems. So my focus for this project is the DAP pathway, the diamino-pymalic pathway, which is shown here, which is the pathway for lysine biosynthesis. Now, I am focused on the enzyme DHDPS, dihydropicolinate synthase, which catalyzes the reaction between aspartate semialdehyde and pyruvate to produce HTPA, and followed by a subsequent multi-step pathway to then produce lysine. Now, once the plant has produced sufficient quantities of lysine, that lysine then goes back and binds allosterically to this enzyme and turns it off, preventing any more lysine from being produced. So what I want to try and do is target this feedback inhibition and prevent it so that we can produce a lysine-insensitive enzyme, and we'll be able to produce a lot more free lysine. So the first thing I did to try and do this was produce some overexpression lines in both a wild type and a mutant DHDPS. I took my wild type and mutant constructs and used particle bombardment to introduce them into the plant, and then grew these up and to maturity to investigate their seeds for the lysine content. So I did this using HPLC, harvested the seeds, grind them into flour, extract the amino acids, and then derivatize it with OPA. I just have all the HPLC conditions shown here. And I just want to acknowledge Slagiana and Becca at the Maze Research in Belgrade for helping me optimize this protocol. So I had many lines for both my mutant DHGPS, what I'm calling the wild type DHGPS, and some controls. And after running many repeats of each of these lines, Sadly did not see any increase in lysine. As you can see at several different places, there's no significant difference between our wild type overexpressor group, the mutant overexpressor group, or the controls, which after going through all that effort was very disappointing. So I'm currently investigating why this might be, what perhaps has gone wrong, or the reasons for this. Could it just be that the transgene is not being expressed highly enough? The protein could be unstable, or maybe the lysine is being turned over or incorporated into proteins immediately, and so when I'm measuring free lysine, I'm not seeing any change. Perhaps, I'm not sure yet. But I also measured some other amino acids of interest, because these are also part of the aspartate super pathway. So when we have aspartate semialdehyde reacting with pyruvate, we have other reactions coming off of that, and aspartate then leads to threonine, isoleucine, and methionine being made. So by trying to increase the amount of lysine, I didn't want to negatively affect the production of these other amino acids, which also showed no change and no significant difference between groups or within individual lines within those groups, which was good, I guess. So while I'm trying to figure out perhaps the reasons for these results, I needed to come up with another approach to try again. So those plants were transgenic, but the aim of my project is to produce a line of wheat which is cisgenic and can align with the PBO regulations in the UK at the minute. So I designed a new mutant DHDPS construct with a substitution shown here, and also based off a paperwork from Yang et al., where they produced rice, which is high in lysine by targeting DHDPS. And there are also 5 copies of the DHDPS gene, 2 on the A genome, 1 on the B genome, and 2 on the D genome. And the copy on the B genome is the most highly expressed in seeds, and so I targeted this copy of the gene. So I then sent that DNA off to be synthesized, bulk up the DNA, and then going to be using particle bombardment again with the help of the serial transformation team here at Rothamsted to introduce this construct into the wheat and grow it up maturity so that I can analyze the lysine content in the seeds again. So that's kind of where I'm at currently in that aspect of my project. However, I also have another chapter in my PhD project, which is investigating the use of DHDPS inhibitors, of which I'm investigating 3, FBDTA, MBDTA1, and FBDTA2. Now, the reason why I'm interested in these inhibitors, which can also be used as herbicides, is that I want to use them as a selection mechanism during the transformation process. So typically, you would use antibiotic marker gene, bar gene, something like that, to identify which plants have been successfully transformed. However, when you have a bar gene in there, that is now going to make my plant transgenic rather than cisgenic, which I'm aiming for. So instead of going through that process where I then need to cross out the bar gene, I want to use DHDPS inhibitor to successfully identify plants which have been transformed. When exposed to these, they will survive, whereas the plants which haven't been successfully transformed, when exposed to these, will die. Therefore, I can use these as a selection mechanism instead of having to use a bar gene and then or another antibiotic gene and then cross that out afterwards. So it hopefully should save me a big step, and I'll end up with cisgenic plants. So these all aren't commercially available. So I had to synthesize these from scratch. It took a couple months. And then I need to investigate their efficacy. So I've done several experiments. This is just one of them here, where each of the inhibitors has been exposed to seeds. The top row It's just plain background cadenza. And the bottom row is line 59, that double null for the low asparagine, low acrylamide wheat, which I talked about earlier. And then I have some controls of water and DMSO. These are at 500 micromolar. And after 3 weeks, you can see the germination with water, it's completely normal, and DMSO, there's been some germination still. And then looking at our 3 inhibitors, hopefully you can see from this photo that there's been little to no germination whatsoever with FBDTA, and I am going to be selecting FBDTA to use as my inhibitor moving forward, as it shows to be more effective than the other 2 inhibitors. But I still need to do lots more experiments with this. I want to investigate how these inhibitors affect my transgenic plants I've produced, and also my cisgenics once I've made those, and do some more quantitative analysis and dose response curves to identify the exact concentration of the inhibitors that is the most effective. So just to summarize everything I've spoken about today, the low asparagine wheat trial is coming to the end, but we have just published the most recent results in PBJ, so please go and check that out. And the Probity project is still ongoing. In terms of my lysine work, I'm currently just finished bulking up my DNA that can now be used to be incorporated into the plant by particle bombardment. And I'm still doing much more investigation on my DHD inhibitors to use them as a selectable marker instead. So I just want to acknowledge everyone that has contributed to this work, primarily Sarah for all her work in producing the low asparagine, low acrylamide wheat, as well as Nigel Halford, who's my PhD supervisor, Navi, the serial transformation team at Rothamsted. A big thank you to Mark Wilkinson for helping me produce my transformants, and the farm team at Rothamsted for their help with managing the field trial, and of course all my funders. Thank you very much.
Yeah, thank you, Tash, for also another inspiring talk and making also our coffee break biscuits and the breakfast healthier. And I'm looking also forward to your progress with the high lysine wheat. So also this talk is now open for questions. Are there already questions in? This is prime editing. So I think here it starts from Julie. And do you have a paper associated with the whole genome sequencing of the VDAs and 2 lines that has segregated away the transgene?
So we haven't published all of the whole genome sequencing data. Navi is still going through that and doing some bioinformatics on that we do hope to publish this year. So that will hopefully be out very soon. Yeah.
Okay, let's see. Are there more questions in the chat? So because I was also wondering, as you mentioned, your cisgenic approach, have you considered also maybe using prime editing to modify the gene in order to prevent the lysine binding? So is the lysine binding domain maybe known and you can interfere with this by methods of prime editing?
Yes, so the lysine binding site is known, and it is allosteric on the DHDPS. And when I've generated my mutants, I have targeted that site, and that is what I've been changing so that don't affect the other function of the enzyme. So I have been targeting that. The original plan for my project was to use prime editing to do this, producing those transgenics and the overexpression line was purely done for a proof of concept to make sure that targeting DHTPS, could work as a right approach. The next step was then going to be using prime editing to do that. Unfortunately, I obviously didn't get the results that I was expecting when I analyzed those seeds on the HPLC. So I kind of had to reassess and change tactic. And hopefully, when I've produced these cisgenics, I'm hoping that I'll see a increase in lysine. And once I know that this mechanism does work by targeting this, then absolutely the next step is going to be using prime editing to do this.
So there is one question from Agnes. So could you explain why your edited lines produced more but smaller grains? So this is for the acrylamide story. So
yes. Honestly, we're not too sure. That's just something that we noticed, but it hasn't affected the yield. And as I said, the plants produced more seeds. They were just smaller, so it hasn't actually, it's not affecting the yield of the plant. As to why that happened, we're not sure. But as it hasn't, had a negative impact on the plants, it's not something that we're concerned about.
Also would be some sort of sync relationship that if you have more grains and still not sufficient assimilates, then we make just more but smaller grains.
Yeah, possibly. Yeah, it's definitely something that we can look into and that we want to. But the field trial is coming to an end now. So we're going to need to find someone to do that.
Yes, yeah. So are there more questions? So Geraint also put here some additional information in the chat. So if there are no more questions, I would like to thank again both speakers, Tash and Tien, for very nice and great talks. And I would like to thank also all the participants and want to invite them to join us again in June. So definitely this will be also another nice webinar. So I wish all of you a nice day and successful experiments, and hope to see you again in the middle of June or in any other place. So have a nice day. And so as I think in Europe today, there's another step forward for getting the NGTs done in Europe, that we can do similar experiments like you can do in the UK. So fingers crossed, and hope to see as many of you again. So many thanks from my side also.